Kelly Shepard and Stephen Lin from the California Institute for Regenerative Medicine, CIRM. Kelly is the Associate Director of Discovery and Translation, and Stephen is a Senior Science Officer. The mission of CIRM is to accelerate stem cell treatments to patients with unmet medical needs. And today, we will hear more about CIRM and the use of stem cells for predictive modeling and therapy.
Thank you very much. Everybody can hear me, I assume. I'm going to be asking for help with advancing my slides because my connection may be a little bit slow. So I'll try to remember to ask for that. Anyway, I want to begin by thanking you for inviting us. Stephen and I are program officers, as Liz mentioned, at the California Institute for Regenerative Medicine, or CIRM, which is a California state agency that was established by a ballot initiative in 2004 to fund stem cell research in California. Primarily, it was established to support the use of human embryonic stem cells, but it actually supports stem cell research in a much wider variety of areas. And we've been funding over 1,000 projects since 2004, over the past 15 years. Is tag team our presentation a little bit today. I'm just going to start things off by providing a bit of background on our activities over the past 5 years or so, since this is more relevant to what we're talking about today. And this will provide some context for how our perspective has been informed by what we see as current issues and needs in the stem cell field. And then after I go over that, I'm going to transition to Stephen, who will provide some specific anecdotes and examples of some projects from CIRM investigators and the relationship to projects that have been done on the space station thus far and how they might be further informed by such studies. Okay, next slide, please. So as Liz introduced, the mission of CIRM is to accelerate treatments to patients with unmet medical needs. And our goal is to use stem cells in any way possible to do this. Next slide, please. Specifically, that means we fund research and development. And we focus our research in 5 core areas, which are depicted on this bridge. Sorry, I forgot to say change slides, didn't I? I won't really be talking much about our educational pillar today, but I will be telling you primarily about some of the work done through our first core pillars here, infrastructure, which is to create resources for the scientific community to use. And then our pipeline programs, which fall under 3 different stages of maturity: discovery, translational, and clinical. So by discovery, I mean the earliest stage of research. And we have supported both from the very get-go, no preliminary data, hypothesis testing, new ideas, figuring out what happens, how to make things work, to candidate discovery, looking for something that can be used as a potential therapy or as a potential tool for identifying therapies, and knowledge generation. The translational stage is where somebody's made a promising finding in the lab, a development candidate, and they need to take it through the steps that is necessary to begin discussions with the FDA about bringing to a clinical trial. And then, of course, there's the clinical stage, which to CIRM begins at IND enabling, which is after you have agreement with the FDA on a promising set of studies that could lead to an IND filing, to phase 1, 2, and 3 clinical trials. And ultimately, we don't have anything at the stage yet, but ultimately, hopefully, a drug approval or commercialization. Next slide, please. So the types of projects that fall into these pipelines are quite diverse, as you can see. They cover a wide range of activities, but they kind of fall down into 3 basic areas. So knowledge generation, using stem cells to understand basic human development and embryology, to understand the basis of human disease, to discover new disease targets. We also support a variety of projects where stem cells are used as a tool of discovery. So for example, to develop a screening platform for drugs, to model disease in a dish, these types of studies. But we don't just fund stem cells as a tool, we also fund traditional tool development that can be used to enable the stem cell field, such as novel imaging modalities, new cell editing technologies, CRISPR-Cas9, these sorts of things, directed towards editing stem cells. So basically funding tools and technologies that can make stem cells more useful. This is we're also interested in developing things that can make automated processes, because ultimately to bring therapies based on stem cells to the clinic, we need to make them more expensive and practical, reproducible, and consistent. And so I see a lot of common ground there in the needs for what it takes to bring a study into space to what it takes to bring a study into the clinic. And so next slide, please. When we think about the types of these projects that I've just shown on the slide built across this pipeline going from the earliest stage to the clinical stage, you're going to see a lot of common ground, as I said, with some of the types of studies that are being conducted in space and being contemplated, and some of the studies indeed that we've heard about today. So at the very early end of the spectrum, some of the most significant questions then when CIRM was founded and now relate to the rationale. If you want to develop an application for a stem cell, what is the right stem cell type to use, or derivative of a stem cell? Do we know what our target cell is? Do we know enough about human biology to know which the right cell is to study or is responsible for a disease? Is it one cell or is it a group of cells together that are necessary, right? So even if you know that cell, how do you procure that cell? A lot of the work that CIRM funds are trying to procure these cells by differentiating them from a pluripotent cell starting point. That's a scientific process of discovery to be worked out on its own. In addition, stem cells can be obtained from tissues, from adult tissues, but being able to study human stem cells, such as those, which have to be removed and propagated in the lab, there are a whole bunch of challenges around culturing and maintaining them in the lab while they maintain their potential and functionality. It's very hard for many adult stem cells to maintain their ability to be expanded or even activated outside of their niche, which is normally in a tissue inside of an adult human organism. So other considerations that we have to think about, of course, even at the earliest stage, are donor source and eligibility. Eligibility, because if you're going to eventually use one of these cells as a therapy, it has to be consented appropriately to get through the FDA approval process and to be ethically sound. Donor source, we have genetic background of the donor that comes into play. This is also important if you're interested in studying disease processes. You want to, as Arun mentioned in his talk, you know, the possibility of taking cells from individuals with specific diseases so that you can model them in vitro. So those types of considerations. As we move along to more mature science, getting into translation or early development and eventually clinical trials, the challenges become quite different. So at this point, you've already identified a promising stem cell-based candidate that you want to eventually test in a more broad use application. Here are the challenges developing, and we heard about this a little bit in our first presentation as well, is the scaling up. You're going to have to go from a dose that might have worked in a rodent to a dose that is going to be effective in a person. So you're going to have to be able to grow the cells in much larger numbers while maintaining their functionality and potential. And not only that, you're going to have to change your process so that it meets GMP compatibility requirements, so that it can be produced in a gene compatible manner. And that can change, that can have effects on your cell. So a great deal of challenge, especially in our field, is centered in this space right here and right now, and that's developing the GMP compatible process and the scaling up process. And although some projects have made it into clinical trials, of course, there are a whole new set of challenges that they're facing and navigating now and will continue to do so. And there's even more scale up that will need to happen if this is going to be something that is going to be offered as an approved drug to a more widespread disease. You can't just make enough cells for one patient. You are going to want to treat a lot of patients. So you have to be able to develop a commercial scalable process. You have to figure out how are you going to store and ship your cells. And all of this, you have to maintain the integrity of the cells. And so we're not quite there for most of the types of projects that CIRM funds, but these are the challenges that we see as looming on the horizon. Okay, next slide, please. So this is the state of the field today. There are a number of stem cell projects that are in clinical stage trials or very close. The more mature therapies that have been worked out longer are the mesenchymal stem cell products. And we also have a long history of bone marrow transplants now, which are basically hematopoietic stem cell transplants. There are also some examples of some primary neural stem cells in the clinical trial, as well as immune progenitors. such as T cells and dendritic cell progenitors, and other tissue-specific progenitor cells. There are also now some pluripotent stem cell-based products that are also in clinical trials. You may be aware of some of these. We have pancreatic progenitors. There are some cardiomyocytes derived from human embryonic stem cells, neural progenitors, dendritic cells, retinal derivatives. The ones with little asterisks are the ones that are currently in clinical trials. The others are getting very close. But as you can see, the field is evolving, and a lot of the technology that was in its infancy 15 years ago is getting ready to a stage where these questions and challenges are a reality, and they need to be addressed now. Next slide, please. So this is just a snapshot of some of the earlier stage projects that CIRM's been funding over the past 5 years or so through our candidate discovery program and our hypothesis testing program, which we call DIS1 and DIS2. So you can see on the left that about half of our projects are dedicated towards trying to find a therapeutic candidate, either from stem cells or that can be discovered through the use of stem cells. We still fund a fair amount of projects doing basic knowledge building, hypothesis testing, and about a third of our projects are devoted towards developing stem cell-based tools or technologies, or, as I said, imaging technologies that can be enabling for the stem cell field. Although the majority of our early stage programs are still focused on pluripotent stem cells, primarily embryonic stem cells, induced pluripotent stem cells, or using both, we do have a fair amount of projects looking at hematopoietic stem cells and a whole variety of other adult stem cell types. And we also have some projects who are looking at direct reprogramming, which is skipping the pluripotent stem cell step and trying to convert one differentiated stem cell type into a different fate, to sort of avoid some of the challenges that come with working with pluripotent stem cells. Next slide, please. As we move down that pipeline, as you can see, our translation stage, we still fund a fair amount of pluripotent stem cells there. Again, these are projects that are engaged in studies that will allow them to bring a package to the FDA for discussion. Now, you can see that a good portion of these are hematopoietic stem cell projects, but these aren't just your ordinary bone marrow transplant. These are gene-modified hematopoietic stem cells. So CRISPR-Cas9 edited or lentiviral modified. So these are kind of the next frontier of stem cell biology. So there are also a lot of challenges to be worked about to get efficient editing of these stem cells so that they can be used in a safe and practical manner. Next slide, please. And this is the last slide I'm going to share before I transition to Stephen, because what I want you to see is that there really is a lot of momentum in our field building for the pluripotent stem cell derived therapeutic products, as well as what I just alluded to, gene-modified versions of established stem cell products, such as hematopoietic stem cells. But as you can see from this kind of green coloring and on the right here, even these darker green colors here, even a lot of the pluripotent stem cell products that we're funding are gene-modified. And as you can see, these are primarily the allogeneic candidates, meaning donor-derived. And as you can imagine, or you might guess, a lot of that gene modification is an attempt to make a universal cell product, something that can be used by people with different immune backgrounds. Now, you've talked a little bit about manufacturing cell therapies in space, and I think that probably makes the most sense for a product where you could produce a bunch of it and bring it back and have it be an off-the-shelf therapeutic. So figuring out how to create immune compatible cell types that could be manufactured safely and effectively and inexpensively is going to be something that's extremely valuable for the field as a whole. So I think from here, I'm going to transition to Stephen now, and he can give you a few more concrete examples of some of what I've just shared with you in our overview.
Great, thank you, Kelly. Can you advance to the next slide? Okay, so as Kelly described, CIRM has established a pipeline of awards that span from basic research and discovery to clinical stage programs. And so I'm going to describe several examples of CIRM investigators that are developing therapeutic products at different stages in CIRM's pipeline, but they've also conducted projects aboard the ISS or are planning to do so. And actually there's some overlap from the previous presentations to some of the examples I'll give. And so I'll also describe some scientific questions related to space research that we've seen arise from the programs that we've supported at CIRM. Next slide, please. So beginning with our discovery stage awards, Sonia Schrepfer at UCSF had a project to develop hypoimmunogenic cardiac patches from iPSCs. And so one important hurdle that has always existed with cell therapies is rejection of the graft by the immune system. So we've had several CIRM projects that attempt to engineer cells to evade the immune system, employing different methods to protect the cell therapy from B cells or T cells and the innate immune system. Next slide. So Sonia Schrepfer also has a project with a board that will go to the ISS. She has a project to look at the effect of immunosenescence from CD8 positive T cells on mesenchymal stromal cells as a model for bone healing, and endothelial progenitor cells as a model for vascular regeneration. So microgravity is a model for aging in this case, and the hypothesis is that in microgravity, there will be an increase in terminally differentiated CD8 positive effector memory T cells, and that can impair stem cell-based health in tissues. So these experiments set up broader questions that you may ask in space, including what is the immunogenic profile in microgravity, or what happens to the immune response in space? Next slide. So Jean Loring at the Scripps Institute in Aspen Neuroscience had a later stage discovery award to make an iPSC derived dopaminergic neuron that's ready for translation. So this is an autologous therapy, and she was doing many of the studies in her CIRM award that were needed to establish reproducibility of the product, including establishing differentiation to dopaminergic neurons for 10 patient lines, determining the functionality of those patient neurons, assessing the immune response of that product, looking at cryopreservation, and then looking at dose response and dopamine release for function in vivo. So a lot of the product attributes that Kelly touched on. Next slide. So in partnership with the New York Stem Cell Foundation and Space Tango, as Janice Studemeyer described, and also the Parkinson's disease patient advocacy group Summit for Stem Cells, several of the iPSC lines that were created from Parkinson's disease patients are going to the ISS. So neural organoids will be created from these from the patient iPSCs to determine how microgravity affects their migration, neuronal changes, and a gene expression pattern. So deriving mature cells from pluripotent stem cells has long been a challenge in the field. And so a broader question is whether microgravity affects the function of iPSC differentiated cells such as these, or could possibly improve them. Next slide. So Joe Wu at Stanford has been supported by several CIRM awards, including a disease team grant to get embryonic stem cell derived cardiomyocytes scaled up and tested for a clinical trial, and a research award to create a bank of iPSCs from cardiomyopathy donors for disease modeling and drug screening. Next slide. And so his group has built a large iPSC collection of cardiomyopathy samples. And this is a project that the previous speaker, Dr. Arun Sharma, described when he was in Dr. Wu's lab, looking at iPSC-derived cardiomyocytes that were sent to the ISS compared to their effects on normal gravity. And he went through the data much better than I could, but there were some differences between the cells that were sent to space and the ones that were left and regular gravity on Earth, including some hints of possible differences in beatings and arrhythmias. So one question is whether stem cell models like this could predict human health with long-term space travel. For example, could they be used as a screening tool to predict whether a person is at higher risk for heart arrhythmias in space? And is there a genetic signature that could be associated with that? Next slide. And so in one final example of a CIRM-funded investigator is Dr. Clive Svensson from Cedars-Sinai Medical Center. He's funded by CIRM for multiple clinical trials using stem cells. One trial involves using human neuroprogenitor line that expresses the neurotrophic factor GADNF to treat amyotrophic lateral sclerosis, or ALS. The other trial uses the unmodified neuroprogenitor lines to treat the eye disease retinitis pigmentosa. Next slide. And so the group plans to send 2 types of cells to the ISS in partnership with Space Tango. The GDNF neuroprogenitor line used for the ALS trial and undifferentiated iPSCs. And both Janice Studemeyer with Space Tango and Dr. Sharma are probably involved in these projects. So one challenge with PSC manufacturing is to maintain the stability of the cells with passage and expansion. So pluripotent stem cells can lose their pluripotency potential, and they can become karyotypically unstable with prolonged culture and expansion. However, expansion is critical for many PSC-derived therapies to set up working cell banks so that you have enough to go into the patient. And so one question is whether these characteristics can be better maintained in space. Next slide. So these are some examples of CIRM investigators that have studied various aspects of stem cell biology in space in connection with their translational and clinical efforts on Earth. And at CIRM, we've encountered many other bottlenecks over the years with projects that we've supported. Kelly has touched on a lot of them, but other bottlenecks that we've seen that perhaps space could address are organoids. When organoids are put together, usually they are still in a fairly immature state. And so the question is, if you assemble the organoids in space, can the self-assembly of organoids create more mature populations or different populations than you could see on Earth? Another challenge that we see on Earth is the bottleneck in expanding somatic adult cells in vitro. So hematopoietic stem cells or liver hepatocytes, they either self-renew or they can regenerate in the body. And that's one of their characteristics. Interestingly, when you take them out and you try to grow them in culture, they act like they're quiescent. They really do not expand. And it has been a challenge for decades to get them to do this. The question is, if you put this in a microgravity environment, can you adjust the conditions to allow them to do this? Also, cryopreservation. So cryopreservation is a significant issue with cell manufacturing for your product. You want to cryopreserve the cells to make the cells more feasible for distribution and transplantation, but there is a drawback in that cryopreservation also often leads to a loss of functionality. And so if you can adjust the cryopreservation conditions in microgravity, one question is whether it could overcome this loss of function that you see on Earth. And then the final bottleneck, there are many other bottlenecks, but the final one that I'll just touch on is genomic stability with iPSCs, which we heard about, which I described at the last example with Cedars-Sinai. So this is a significant issue with cell manufacturing, and it'll be interesting to see with this Cedars project if microgravity can improve that clonal stability. Next slide. So another recurrent issue that we see is that phenotypes can vary significantly among cell lines. So one cell line may respond very strongly to an experimental condition, and another may not have any effect at all. And so with PSC lines, some PSC lines differentiate to certain tissue types better than other lines, and it's really not known why. So looking at cell line variability is very difficult aboard the ISS because, as was described very well by Jana Sudermeyer, there are very tight confines with these experimental cabinets. So in addition to funding research investigators with the examples I described, CIRM has also created resources that exist that assist the entire scientific community. And this is part of our infrastructure pillar of programs. So one of the resources that we've established is an IPS bank. And this IPS bank is currently the world's largest collection of publicly accessible iPSCs with over 2,600 lines. It's also one of the most diverse, with lines represented from African, Hispanic, East Asian, South Asian, and European ancestries. So the cell repository is managed by Fujifilm Cellular Dynamics. And several years ago, CIRM and Fujifilm began a partnership with the Broad Institute in Massachusetts to utilize the breadth and diversity of the collection to create pools of stem cell lines termed cell villages. Next slide. So in the system that the Broad established, up to 100 lines can be pooled in a single culture and later stored for a specific phenotype. So using natural variations in genomic sequences as barcodes, individual cells can be identified that have low to high phenotype characteristics, and a new analysis technique that they developed called census-seq can profile rare alleles that are associated with the phenotype. So the proof of concept experiments for this approach map the effects of several alleles on drug responses for spinal muscle atrophy. And so because this technique enables population scale experiments in very small pools with high internal control, one could see it being utilized eventually aboard the ISS to examine genetic effects of diverse phenotypes in a confined compartment. Next slide. And so this is the final slide that I'll go over. As a funding agency, CIRM wants to support stem cell technologies to advance its mission. And if there are organizations in CIRM that are in California that are developing tools that utilize stem cells for the ISS, the agency has a single funding opportunity this year to support it. As Kelly talked about, Discovery Tool Awards will support tool discovery that will demonstrate technical feasibility in a research prototype. So the tool must use human, stem, or progenitor cells, but otherwise it's fairly open what you can apply for. This award is $250,000 for 12 months. And if you're interested, the deadline for these DIS2 awards applications is August 31st. And more information can be found on CIRM's website, cirm.ca.gov. Next slide. So if there are any questions, please feel free to ask Kelly or me at the information on the slide. Thank you very much for your attention.